The dorsal root ganglia contain all sensory neurons that represent the body. These neurons receive signals from the musculature and skin and transmit them to specific laminae of the spinal cord. The perception of temperature is mediated primarily by sensory neurons with A delta and C class axonal fibers. Researchers discovered that sensory neurons that respond to heat are also activated by capsaicin, the active component of a chili pepper, and that sensory neurons that respond to cold are activated by menthol, the active component of mint that gives the sensation of coolness. This phenomenon was used to identify the specific membrane receptors responsible for sensing temperature. Using a technique called expression cloning, researchers transfected pools of cDNAs derived from dorsal root ganglia into cultured cells. They applied capsaicin to see which pools of cDNA would produce an intracellular calcium rise in response to the application of capsaicin. After repeatedly dividing the cDNA pool into smaller and smaller pools, a single cDNA from the pool encoding TRPV1 was identified as producing the intracellular calcium rise. mRNAs encoding TRPV1 were injected into xenopus oocytes so that they would express the TRPV1 receptor. Researchers then used a 2-electrode voltage clamp to record the current flow across the oocyte membrane in response to experimental conditions. Researchers found that TRPV1 is activated by both heat and capsaicin, as indicated by the production of the inward current in the voltage clamp setting. A similar experiment using menthol in place of capsaicin identified TRPM8 as the cold-sensing receptor. Injection of mRNAs encoding TRPM8 into a xenopus oocyte confirmed a response to cold and menthol, but not to heat and capsaicin. When mRNAs encoding both channels were injected, xenopus oocytes respond to all 4 stimuli: cold, heat, menthol, and capsaicin. Thus, these 2 TRP channels, TRPM8 and TRPV1, allow animals to detect cold and heat temperatures. Since these discoveries, additional temperature-sensing receptors have been identified that cover other temperature ranges.